All right, so I'm on page 84 of your science notebook, and you hopefully have already taped down all of your cells in order, and I'd like to go through some details that I need you to add to your cell cycle and mitosis note sheet. So way up at the top, it says that we're making more body cells. I'd like you to make sure that you understand body cells. When we talk about, we're very, very focused on humans. So we're going to talk about skin cells, or we're going to talk about muscle cells. We're going to do this with any of our connective tissue cells, our fat cells. The cells that typically don't go through this process are things like nerve cells. That's the reason why we like you to be very, very careful with your brain, because once you've damaged those beautiful nervous system cells, you don't really get to regrow or regenerate these. Those are some of the few cells that last throughout your entire life. So your body cells, you've got about 200 different types of body cells, and when they are tired, damaged, or just old, they will die off. It's a process called apoptosis, which is pretty cool because it sounds like the cells are going to pop, and that's almost what they do. So before that, other cells have to go through the cell cycle and mitosis to make more, to replace the ones that are old and needing to be taken out, kind of like they're spent cells and they need to be trashed out. So this is the process. The first step is going to be step number 7. This is just a typical cell, regular body cell. You can see its cell membrane, its nucleus has got loose DNA. This is going to be specifically for humans. We're going to talk about human cell cycle and mitosis. So next to this cell number 7, you're going to write that it has a full set of 46 chromosomes, and they're loose in the nucleus. When they're loose in the nucleus and they look like spaghetti noodles, we call it chromatin. Okay, and then we'll see the first true step of cell division. So this is where your cell spends almost all of its lifetime. Every once in a while, the cell will kick into a cell cycle and mitosis moving forward process. And when it does that, then we start with something called interphase. And I'll mention that at the end. So the first cell, you'll notice that this is a bigger cell. And we're looking at this nucleus that looks a little bit different. So this first cycle is called the G1 phase. We call it the gap phase. Sometimes we just call it growth phase also. But you'll notice that the cell is bigger. And it's bigger because the cytoplasm has doubled, and some of the organelles that live in this cytoplasm have also doubled. So we'll have more mitochondria, more chloroplasts. So next to this third cell, I'd like you to write cell growth. This is the cell number 3. And it's called G1. Then take a look at the next cell cycle. The next cell phase is called S phase. S phase stands for synthesis. Sorry, I'm having trouble with this. Synthesis. Synthesis means to make something. And if you take a look, the cells are the same size, but what you're noticing is there's a difference between what's in the nucleus. These are single little pieces of DNA, and these have a left and a right side. That means that DNA got replicated during this S phase. DNA replication is when the single strand of DNA cuts open at the nucleotides by helicase. New nucleotides are put on each side, and then DNA polymerase can add those nucleotides and proofread, and ligase comes in and fills those gaps. And then you have 2 completed exact copies of the DNA. So you have a left side, let's see if we can zoom in. You'd have a left side, and then you would have a right side. And these 2 are joined at the middle. This is called a replicated chromosome, okay? So this is your S phase. DNA has been copied. Next phase is another growth phase. So look how much bigger that cell is. That cell is significantly larger. The DNA is still doubled in the nucleus. So we've got the beginning of this cell cycle. G1, S, and G2 are all part of something called interphase, and we'll label that at the end. Next, we're going to get into the actual process that divides the nucleus. So, cell 8 is the next. That's prophase. Prophase can be described as the phase where the DNA tightens up and is really visible as chromosomes, and the nucleus dissolves. You can see the two sister chromatids. You could see them before, but not really. This is just for our model. In real life, you can actually start seeing the DNA crush up real tight and become chromosomes at this phase. Then you've got metaphase. Metaphase is when all the replicated chromosomes line up in the middle. So over here, we're going to write replicated chromosomes line up in the midline or middle. Then we get anaphase. Anaphase is when these long fibers pull apart the replicated chromosomes and they go back to opposite sides of their cell. So we'll see chromosomes, two sister chromatids, get pulled apart. Sometimes we call this anaphase because it sounds like the chromosomes might yell when this happens. Next is cell number 9. You can see that we've got all of our chromosomes over here getting wrapped up in new nuclei, and the cell is pinching away and starting to divide into two. So over here, telophase is the original cell starts to pinch apart and new nuclei are formed. And then the last one, you'll see ... because this is the end of mitosis. And the last step is cytokinesis. Cyte means cell and kinese means moving apart. So you'll see the division is finalized. The nuclei are all back and formed, and the DNA becomes loose chromatin again. So this says finished, 2 exact cells, both with 46 chromosomes. So these 2 cells are finished and the division process is done.